We're going to do meiosis is the topic. If you don't study anything having to do with organisms or genetics, finding a paper that's relevant to your topic and meiosis might be difficult, I understand. Do your best. Spend 3 minutes or less looking for a paper. Hopefully you'll find one that's relevant to you and to this topic. Maybe not. So, trisomy and triploidy are corrected during female meiosis and C. elegans. So basically what I want to do in the next half an hour or so, maybe, is to remind you about meiosis, in case you've forgotten or never knew it in the first place, and then also cover a few key topics like trisomy and triploidy. You may be familiar, but again, I want to make sure everybody's on the same page. So, we already talked about ploidy today, so this should go pretty quickly. Humans are diploid. Okay, so we've got diploids are 2N. They have 2 copies of every chromosome. What other sort of ploidy do you know about in any sort of organism? Haploid, which we just call N, right? So what sorts of cells are haploid? So some yeast are haploid, gametes of diploid organisms are haploid. We've got organisms that are tetraploid and all sorts of ploid. So there are some salmon, lots of fish are tetraploid, and then you get into plants and they do all sorts of weird things. Like some strawberries are dodecaploid. They've got 12 copies of every chromosome. Yeah. Okay, so that's the term ploidy. We're into ploidy for several reasons. This is one. We should be interested, just as humans, that there can be changes in ploidy that are naturally occurring. Right? Spontaneously, some frequency of individuals, and by the way, I grabbed the wrong karyogram. So this is a karyogram of trisomy 18, where an individual is diploid for every chromosome. There's 2 copies of every chromosome, except for 18, where there are 3 copies here. Right, so trisomy is the nomenclature. So triploidy would be 3 copies of every chromosome. Triploidy. Trisomy is when a single chromosome is aneuploid, has the wrong number of chromosomes. So Down syndrome is actually trisomy 21. And an important but subtle point to make here is that you can't be trisomic for every chromosome. Humans can, at least. So the only trisomies we know about are 18 and 21. What do those chromosomes have in common? Of all of the chromosomes in humans, 18 and 21, are they big numbers or small numbers? Right. And human chromosomes are ordered by order of size.
Oh, yeah, so they're small.
So 18 and 21 happen to be some of the smaller chromosomes. It turns out that we think that the reason that trisomy 18 and 21 are the only known trisomies that are compatible with life in humans is because those chromosomes don't have as many genes. They were less likely to have genes that are dosage sensitive, where it really matters for life if you have 2 copies of a gene or 3 copies. So you can't be trisomic 1, you can't be trisomic 2. Just because those chromosomes are bigger, they've got more genes, there's more likelihood that having an extra copy of the chromosome will really, really severely disrupt development, causing death. So trisomy 18, trisomy 21. Biologists in general, some molecular biologists are interested in how trisomies happen. And you might know that there's, for example, in human females in meiosis, there's an age-related onset of increasing problems with meiosis that cause gametes to have more or less copies of a chromosome than they should. So that's another reason to be interested in this topic, potentially. So we already drew mitosis when we were talking about yeast a little bit ago. So I want to talk about meiosis. So you start with a diploid cell. So each black horizontal line would be 1 double helix. So 2 copies of this chromosome. And then what happens during meiosis? First step in any sort of cell division is synthesis. So we're going to go from 2 copies of this chromosome to 4 copies. Now, what's the difference between mitosis and meiosis is there are 2 rounds of division. So the first round of division, by the way, we're going to draw in pairs of homologous chromosomes. So the first division, the 2 homologs divide into daughter cells. And by homolog, I mean, you may already know this. You might consider that this is the version of that chromosome you got from your dad or paternal, and there's 1 copy of every chromosome in every cell that's maternal. So that's what's indicated here is both of those chromosomes get copied. So you get 1 gamete that has both copies of the paternal version of that chromosome and another daughter cell that has both maternal copies. Okay, so far? And then the second division, so that's division 1. Second division results in how many cells? 4 total, where each of those homologs segregates so that every single gamete has 1 copy of each chromosome. So we've gone from 2N to N haploid gametes. That's how Mendel tells us meiosis should work. 1 copy of each chromosome goes into every gamete. That's how it should work. Now we're going to study next week how it goes wrong and maybe how an organism can fix somehow meiosis when it goes wrong, which is bizarre and crazy. So that's basis. Do you know how that changes depending on whether you're looking at male or female meiosis?
Yes.
What?
What does it?
So, well, I guess we better be clear on the question. How does meiosis change regardless of if you're looking at a male compared to if you're looking at female meiosis?
Is it when it's arrested?
That's why it's arrested.
2, not 4. It's the very last step.
Close, yeah. So let's look at male versus female, right? So male, female.
So the male will just have like 2 X and Y.
I didn't mean sex chromosome differences. I'm sorry, I should clarify. I mean, when gametogenesis happens in a male versus when it happens in a female, there's an essential biological difference in how meiosis works. And here it is. You start with a 4N after synthesis. You've got a cell with 4 chromosomes. It does the first division. You get 2 diploid chromosomes. It does the second division, and you get 4 haploid chromosomes. So this is what I just drew on the previous slide. So every sperm cell contains 1 copy of every chromosome, in theory. That's what, ideally, that's what happens, sometimes not. In a female, you start with the same scenario, 4 copies of each chromosome after synthesis. But then, something weird happens at the first division. It doesn't look like the male side.
Is it that because it's like I think it's that why it's arrested in the ovaries, and then when we actually, hit puberty, then it's when they start working again, something like that, and when they actually divide again.
Well, there's that too, but that's also interesting. That's not this though.
Isn't the gene?
Size difference. Right. Males, with every single meiosis, a male produces 4 sperm. With every single meiosis, a female produces 1 oocyte. So, after the first division, you get segregation of most of the cytoplasm of the cell into one of the 2 daughter cells. So it's unequal sizes at this point. This is called the Now this, I was mentioning earlier, this is me going off script, so now I'm forgetting what this is called. Primary, the daughter cell that's not a daughter cell, that doesn't become the oocyte, is called the, you'll come back to me. It's a primary something. So then each of these will divide again, and you get segregation of most of the cytoplasm, again from the cell on the left, into one cell. So it's basically unequal cytokinesis. The mature oocyte winds up having one copy of each chromosome, as do the polar bodies. Polar bodies. Primary and secondary polar bodies. Okay, so PB. So you wind up getting, actually I don't remember if the primary polar body divides again or if it does not. The point though is, regardless of that biological distinction, the oocyte winds up getting most of the cytoplasm, but it still should just have one copy of each chromosome. The polar bodies get the other copies of the chromosome, they just don't get used in fertilization. So there's an asymmetry in meiosis between males and females. And that can cause some weird things to happen, some of which may be relevant paper next week. So if every individual cell gets the same number of chromosomes, so we've got a female over here with an oocyte and the polar bodies, and if we've got the results of meiosis from a male with one copy of every chromosome, right, we already talked, well, we didn't talk about it. Mendel's law, one of them is that basically every gamete will get one copy of every chromosome. It's a law. It's sometimes broken, which we're going to see next week. The other really important rule to appreciate for next week's paper is syngamy, which is the fusion, it means the fusion of gametes, the coming together syn of gametes. Which basically means with this sort of, actually let me color code my chromosomes, just for completeness. Let's say dad's got red chromosomes and mom's got blue chromosomes. Same species, just color coding where the chromosomes are coming from. What is the genotype, or what chromosomes are going to be in the offspring? Guarantee, when this syngamy happens, when a sperm and an egg fuse, what chromosome colors go in the offspring? You get one chromosome from dad, one chromosome from mom. So you get one red from dad. Right, so that cell fuses with that cell. So you get one red chromosome, one blue chromosome. Right. That's fertilization. That's how we get from N, or haploid gametes, to a diploid 2N organism. Okay, so that's how meiosis and fertilization are supposed to work. And note, of course, that this brings us back to diploid, no aneuploidy, no triploidy, etc. The organism that we're using next week, so that's just background. I want to make sure everybody's up to speed. Next week, the paper's on Caenorhabditis, which is this genus of nematodes, which happens to be a genus that I study in my research lab, which is why I picked this paper, yes. So I want to give you a brief background on the organism, because this is really going to be important, at least a little bit, for understanding the paper next week. In the species that we're going to be looking at next week, there are two sexes. They're diploid. So this is a diploid species. Every individual has two copies of every chromosome. The sexes are male and hermaphrodite. So the males make sperm, the hermaphrodites might make their own sperm, and they make their own oocytes, or eggs. So the hermaphrodite individuals can self-fertilize and produce their own offspring by themselves. They don't need males. It's really awesome, for many reasons. It makes them a great model genetic organism and molecular biology organism. They're easy to keep in the lab and so forth. Come by sometime, I'll show you some worms.
How do they decide to become like an egg or a sperm?
So this is important. Good question. So the males only make sperm, but when we focus on the hermaphrodites, I am a really horrible artist. That's meant to be the head and the tail of a worm. The hermaphrodites have a structure in the middle of the body, the vulva, which is basically an opening in the body wall of the organism that's next to the uterus, and they've got a long tubular gonad that's got 2 arms. So basically, each of these things is a tube that starts there in the body and goes down towards, on one side of the body, the head, and then that tube continues down and opens into the uterus next to the vulva. And so these worms, hermaphrodites, can either be mated with by males, so males can inseminate hermaphrodites to create cross progeny, hybrids, or they can use their own sperm that they made to produce their own. They do both. They can be mated with by males, or they naturally produce their own self-progeny. And it's weird, yeah. So there's, at the end of each gonad, there's a single cell, the gonad stem cell. And early in life, that gonad stem cell, like any good cell, divides, puts off daughter cells. The first cells that the gonad stem cell, the daughter cells, when it divides, produces gametes. And the gametes travel down this tubular gonad, and I'll tell you what happens next. The first gametes that are produced are sperm. And then later in life, somehow, which we don't know how, that stem cell divides, makes a sperm, divides, makes a sperm, divides, makes an oocyte, divides, makes an oocyte, switches, same cell, one cell in the body, making sperm, making sperm, making sperm, something changes which we don't know what it is, we don't know what happens, triggers that cell to start producing gametes of the opposite sex, oocytes. Bizarre in itself. But this is just the biology you need a little bit of to understand next week's paper. So those sperm are really tiny. So I'm going to color code the sperm, I don't know, green, I guess. I think that's green, right? Is it green?
Yes.
Okay, good, thanks. So the sperm are tiny, and the sperm, after they're produced, remember they're the first gametes that are produced. They travel down the gonad and they wait here in the spermatheca, which literally means sperm room. It's a little place in the body of the hermaphrodite where the sperm basically wait. This happens when males ejaculate through the vulva of a hermaphrodite, fertilize the hermaphrodite. The male sperm will go into the spermatheca and wait. And the hermaphrodite's own self-sperm do the same thing. They just hang out here, adjacent to the uterus. And then later in life, when those stem cells start making eggs, in blue, let's say, those eggs then travel down the gonad, and what do? They're actually basically being pushed down this tube. So what does an oocyte do? It runs smack into the spermatheca, which is full of sperm, so the oocytes can't help but be fertilized. And then the hermaphrodite deposits the fertilized oocyte, which kind of looks like a round ball with a nucleus, or kind of like a square almost with a nucleus, deposits the fertilized oocyte through its vulva onto whatever substrate the worm's living on. So it's basically an egg at that point. It's a fertilized egg. It's got a shell around it. The worm inside, the larval worm develops, hatches, next generation. Okay, so that's the hermaphrodite. And then there are also males, which are normal males. They just make sperm. They can fertilize hermaphrodites or not. So next week, you're going to see the authors are going to take advantage of the opportunity to be able to set up crosses between males and hermaphrodites. But remember, the title is about triploids. And normally, sexual reproduction in a diploid organism will make diploids. So somehow, these authors have to figure out how to make triploid organisms to study. So how do you make a triploid? Turns out, they can take a diploid male and after meiosis, he produces what sort of ploidy gametes? Sperm? They'll be haploid, one copy of every chromosome. There are also, strangely enough, tetraploid worms, the same species. So somebody made a mutant that has an extra pair of every chromosome. So you take a hermaphrodite from that strain, and it's, what did I say, tetraploid. So after meiosis, its oocyte is what? So if you start with a tetraploid, its oocyte will be diploid, 2 copies of every chromosome. Because going from a somatic cell to a gamete, you always reduce the number of chromosomes in half. So a diploid becomes haploid, a tetraploid gives a diploid gamete. So if you have syngamy, the fusion of sperm and egg from these 2 strains, you make a triploid. Okay, so it was known this is part of the introduction, but it's reference to old papers you're not going to look up or maybe even be able to find. That it's known, so here's the really weird thing. If you've got a triploid, this is the last thing really I think I'm going to do with you tonight. This is a triploid. How does gametogenesis work in a triploid? So if we start with this cell and do meiosis, what's going to happen? It's really weird. So, well, the first step, I guarantee, is going to be synthesis. So we're going to have each chromosome gets replicated. Okay. Then what's going to happen at the first division? Normally, 2 chromosomes would go one way, 2 chromosomes would go the other way. So you're going to have, presumably, you'd have 2 pairs of chromosomes go in one daughter cell and one in the other. And I'm referring to my cheat sheet just so I make sure I get this right. But this is a polar body. I drew it a little bit smaller. There's another thing that could happen here. What's that? There's a second choice for division 1. Right. So you could also get the presumptive oocyte getting the single pair and the polar body getting the extra copy of the chromosome. Right. So what I'm trying to do here is set up for you the expectation of what the experiment should result in that you're going to read about next week. Is if you have a triploid female and her gamete undergoes meiosis, what are the options? With which frequency, or with what frequency should each of those 2 paths, let's call them A and B, which is more frequent? Is it more likely that A happens or B happens, do you think? Okay, we'll go with that. So let's assume that A happens more often. It turns out that the authors are going to claim that these are equally likely. But absolutely, you can hypothesize that A is more frequent. You think that because the oocyte's bigger, and so it's more likely to get an extra copy of the chromosome? I don't know. Maybe. So then, what is the outcome of the second division? This is where the sister chromatids separate. So you'd get an oocyte that has 2 copies, a polar body that has 2 copies, and then that original polar body divides in half. So you wind up getting an oocyte that's got 1 extra chromosome. What happens in the other path B after the second division? The oocyte and its secondary polar body are now normal, and maybe the resulting polar body is diploid. So if these 2 pathways, again, I respect the hypothesis that A might happen more frequently than B, but the authors are claiming that A and B are equally likely. So if that's true, then what happens when you mate lots of offspring resulting from meioses? So some oocytes have this extra copy of each chromosome, and some are normal, 1 copy of each chromosome. You should see how many normal offspring and how many triploids. So here, one more point. We need to make this individual with a male. So he was diploid, he produces haploid gametes. So if these haploid normal sperm fuse with that gamete, you get a diploid or triploid. Right, you get 2 of mom's chromosomes and 1 of dad's chromosomes. But if another sperm fuses with the normal oocyte, you get a normal offspring. 1 of mom's chromosomes, 1 of dad's chromosomes. So the authors are going to tell you, you should expect in this experiment half of the offspring to be diploid and half to be triploid. It turns out in C. elegans that a couple of decades or several decades ago, somebody did this experiment and discovered that you get way more than 50% of the diploids. Right, so expectation, just based on statistics, if you make an infinite number of oocytes and you have these sperm, of those infinite number of oocytes, half of them should be diploid oocytes, half of them should be haploid or normal oocytes. You should get exactly 50-50 triploids and diploid offspring. But they observed that that's not the case. More normal, wild-type diploid offspring than you'd expect by chance. So the paper next week is following up on this initial observation to try to figure out, and hence the title of the paper, is aneuploidy fixed, corrected, during meiosis? Can the can the process of meiosis be skewed, tinkered with, directed, so that non-random segregation happens in meiosis so that you get more of path B happening, where you get the extra copy of the chromosome goes into the polar body as compared to what you expect, as opposed to what you expect in path A, where the presumptive or eventual oocyte gets the second pair. They're arguing that somehow a worm or meiosis or some sort of cellular machinery can force path B to happen more frequently in a triploid mother, so that she winds up getting more normal offspring than you'd expect by chance.